So I've got 15 minutes to update you on the Humanized Bioartificial liver. I need to disclose that I'm the founder of a company called Liver Cell Therapies that has the rights to the technologies that I'll be discussing, both the bioartificial liver based on pig hepatocytes and a bioartificial liver based on human hepatocytes produced in a genetically engineered pig. So many of you are familiar with this field of liver support devices, but the goals of therapy are to detoxify, provide synthetic function, correct this systemic inflammatory response of SIRS, stimulate regeneration of the liver. If all that works out, you'll improve survival. You'll bridge a patient to spontaneous recovery without a liver transplant if the liver is capable of regenerating. If not, hopefully bridge the patient to a successful liver transplant. I'm talking about acute liver failure primarily in this talk. People are familiar with the MARS system, which has shown sort of benefits in some situations, not much improvement in survival, but probably some improvement in hepatic encephalopathy. If you think about this system, remove charcoal, remove the ion exchange column, but leave the albumin in the system. That's essentially what the device I'll talk about is. It's a albumin dialysis system that's had the charcoal and the resin columns removed, and a reservoir with a very large number of hepatocytes, 300 or more grams of hepatocytes for a clinical study that are viable primary hepatocytes, metabolically active. We're not talking about a tumor cell line that may be partially active. These are primary hepatocytes. And this is what the reservoir looks like. The functional unit are these spheroids of hepatocytes that are roughly 100 hepatocytes in aggregate mass, measuring about 75 microns in diameter. We've optimized this process over the past 5 to 10 years, so it's very reproducible that we have spheroids that look similar to this, that then are stable in culture for days to weeks. We've had reports in hepatology of 4 to 6 weeks in culture. The purpose of the device is really as a daily 24-hour treatment. We have a system that we rock these cells. We isolate hepatocytes from a pig, take isolated cells. Each one of these trays is 1 liter of media, 100 grams of hepatocytes. A gram is 10 to the 8th cells, so we're talking about 10 billion cells per tray. So we're talking about huge numbers of cells, 400 grams total isolated from a pig. Over 24 hours, they spontaneously form spheroids. This was a serendipitous observation. One night I put cells in a rocker just to think I could improve the oxygenation of the system, and the next morning I came in and the flask was full of spheroids. So we've patented that observation. But the main thing is you have normal liver functions at a very large dose. Because of the suspension culture system, you have anchorage-dependent hepatocytes that would normally die in suspension, but because they're forming these aggregates, they can stay viable at very high doses. When we perfuse the system at a very rapid rate, I'll show you the slide, the oxygen level comes in at 500 torr and leaves the system at about 150 to 100. So there's a huge amount of oxygen consumption by these very active hepatocytes. This is a slide I have to attribute to Dr. Vilstrup at one of these sessions, and this is why going to social events and meeting outside of the conference room is important. He suggested to me, did you know that if you change the diet on your animals, you can improve their ammonia detoxification ability? And I'd never heard that before, but this is well-established information from the 70s. So at baseline, pig hepatocytes in the device will drop in ammonia from 800 down to about 550 in 1 hour. If you put the pigs, instead of a 15% protein diet, on a 40% protein diet, you can almost triple that. So in 1 hour, you can take it down from 800 to 100. And this is ammonia detoxification with the production of urea through the urea cycle. I don't have time to show the assay, but just feeding high protein diets to our donor pigs has made a big difference in their metabolic activity. So this is the original prototype device that we called the CART. We've actually sold this to a hospital in Chengdu, China that's also working with us on the plan to commercialize or clinical use of the device. This is the current generation device that we're using that I'll talk about, and we're in the process of building a clinical use device that the FDA, we're working with the FDA on approval. So there'll be the clinical guidelines based on that system. The RELF study that I'll tell you about is based on this system. We just recently finished it after my talk today. I'm locking myself in my hotel room and completing the manuscript that'll hopefully be submitted this week. So this is all brand new. It's sort of every day, every week, things are changing. So this is the RELF, Reversible Acute Liver Failure Ambulatory Porcine Model. We use D-galactosamine as our liver failure agent, a single dose. A week before infusion, we do a surgical procedure where we do a baseline liver biopsy. We place a double lumen tunneled catheter, a palindrome catheter, standard hemodialysis catheter, and we place an implantable ICP monitor that allows ambulatory ICP measurement. Give the drug at time 0, follow them 90 hours. Essentially, we give the drug on a Monday morning and we follow them until Friday afternoon. So it's practically the lab staff like that, even though they're there night and day for 4 straight days. So these are very labor-intensive studies. Each experiment is $10,000 to $20,000. So we sample blood, examine the animals. We have a careful encephalopathy assessment that's done regularly, and then we measure ICP by this telemetry technique. At 24 hours, we isolate the cells. And the treatment is a 24-hour treatment starting at 48 hours. And this RELF study includes 18 animals, 6 treated by the SRBAL, 6 treated by essentially a no-cell albumin dialysis circuit, and 6 no-device controls. These 6 were broken down into 3 treated with a Fresenius membrane and 3 treated with a Mars membrane. So essentially a Prometheus system compared to a Mars albumin dialysis system. And cut to the chase, the Mars membrane worked better. There was a slight difference. And then at the end of the treatment, the study is terminated at 90 hours. So the survival endpoint is 90. It's just histologic evidence, D-galactosamine caused liver failure, the brain swell. And this is our Ralmedic implantable ICP monitor. So we place this implantable catheter a week before the drug's infused, so everything heals up, incisions closed, and this is a awake pig on propofol during the 24-hour treatment. Afterwards, the pig's allowed to wake up and walk around. The endpoints, this is based on our Animal Care and Use Committee, survival to 90 hours, and they won't let the pigs die. We have to reach a death equivalent, which is essentially grade 3-4 coma, non-arousal to an ear tug or sternal rub, or inability to maintain sternal posture at an ICP over 20. Those are death equivalent endpoints in this ambulatory model. At the baseline, which is 48 hours after the drug infusion, that's when they randomize to the 3 treatment groups. Essentially, this figure shows that all 3 groups are equivalent. If anything, the hepatocyte group, the SRBAL group, was slightly sicker with a slightly higher ICP, but essentially it says they start off equivalent. This is the system at the bedside of a pig. It's our reservoir, 250 grams in this particular reservoir. The device is perfused at 250 mLs per minute, which is a similar perfusion rate to the portal blood flow through the liver. In order to maximize the benefit of all these hepatocytes, you have to perfuse it at a rate that makes sense, so you're not mass transfer limited. Simple engineering analysis. So we're flowing at a very high rate with a very large number of cells in our albumin circuit. This is a typical control pig. They develop intracranial hypertension and herniate just roughly 65 to 70 hours on average. If you give a 24-hour treatment, you can correct the ICP, the ammonia rises. In the control group, the ammonia is stabilized. This is very reproducible. And that's the benefit of the treatment. The proof is in the pudding. This is a pig up walking around at 90 hours after the BAL treatment and this toxic dose of drug. If you look at ammonia levels in the reservoir, that's in the system, they start out at 0. In the albumin dialysis group, they rise very quickly. This is a 24-hour treatment. They're off the chart, essentially. Pigs that are very muscular animals, have very high ammonia turnover. They're a great model for a study like this because they produce so much ammonia. as opposed to the treatment group where the device is metabolizing ammonia, converting it to urea very rapidly. If you actually do, so it's highly significant benefit of therapy. You do measurements of ammonia production in the two groups, there's a highly significant difference. And then after the treatment, we measure the ammonia concentrations. It essentially goes to zero very quickly at the end of a run. So these cells are very active. The viability starts at 98% and remains 98%. If you measure oxygen coming in, oxygen going out, it's very stable during the 24-hour treatment. There's essentially no loss of function of the cells. This is the survival curves. So in the treatment group with the SRBAL, we had one pig that died, an earlier treatment with the Fresenius membrane. Since we went to the Mars membrane, we haven't had any pig deaths. All of the controls without a treatment were dead at 90 hours. There was one survivor in the Mars treatment group without cells. So this is highly significant survival in this ambulatory model. If you look at ICPs, they start out similar at the beginning. The peak ICP are similar. The one pig that died on the Fresenius membrane had an ICP of 60 when it herniated and died. If you took that out, that number would be down here. So that one animal skews things. But at the time of death, there's a significant ICP difference based on our measurements with this telemetry-based ICP monitor. If you look at brain edema at the end, 82% water versus 80% water, which has a significant p-value, if that means is an important endpoint. Chris Rose helped us with reactive oxygen species measurements. In the control animals, you see a very sharp rise. The treatment's 24 hours, so it continues to rise. ISRBAL, there's an improvement. Even in the albumin dialysis group, there's some benefit of that therapy with regards to reactive oxygen species. So we call this, the FDA calls this our pivotal preclinical trial. Improved survival, duration of therapy, longer is better. We did some animals with 6-hour treatments. 24-hour treatment was much better. They were hemodynamically stable. The longer they were on the machine, the more stable they were. Dose effect, the more cells we put in the machine, the better outcome. If we put in less than 100 grams of cells, it was almost similar to the albumin dialysis group, as you'd expect. Over 200 grams to 300 grams is essentially where we're going to try and go with this for a clinical trial. And this data has been included in an IND submission material to the FDA. They've responded to us. We have a couple things that we need to do. The pigs will come from a barrier facility. These studies were done with just farm pigs, but in order for this to be used in a clinical application, we have to follow certain FDA guidelines. So all the pigs will be raised in a facility that we actually have close by, so it's quite convenient for us. The rest of my talk is going to be about the future, which is sort of the humanized. This is a device now that we've submitted the packet to the FDA, so we're moving ahead with a pig device. The rest of my talk is the holy grail is how do you get an abundant high quality supply of human hepatocytes. And the idea first came, there are a lot of different ways to do this. You can cut a liver in half and it'll regenerate very quickly, as we all know. But you can't take hepatocytes out of a liver and put them in culture and get them to repopulate. So the idea Marcus Grumpy, who's my collaborator on this, had was let's genetically engineer an animal, and he started with mice that are incubators for mass production of human hepatocytes, which sounded almost sci-fi before I saw this paper in Nature Biotechnology. So he's got a triple knockout mouse. There's a metabolic defect and then 2 immunodeficient defects, so the human cells aren't rejected by these mice. And there's a protective drug that fits into all this. But essentially, you inject human hepatocytes, and by day 50, the mouse's liver is repopulated with human cells. If you look in the blood, they're all human proteins. His company, Eucurus, sells a $2 mouse essentially for $5,000 per mouse. Our pigs are a 1,000-fold scale up. The goal is to scale this up to pigs. So that's our hypothesis that we can do this in a genetically engineered pig. Why? Simple cost analysis. If one pig has 300 grams of hepatocytes, which is 30 billion hepatocytes, and the market price currently is $50 per million hepatocytes, a pig's worth $1.5 million at current prices. So our first hypothesis was to make the pig, and we just finished this and published this a couple months ago. So we have the genetically engineered pig. When we started this project a few years ago, nobody had ever developed a genetically engineered pig. The technology for cloning mice is well worked out, but pigs and higher mammals, it's much more complicated. So this is the first genetically engineered pig that has a metabolic liver disease. This is our first litter of pigs. They don't make FAH protein. FAH is important in tyrosine metabolism. Human babies with hereditary tyrosinemia type 1 develop acute liver failure and die very quickly. So there's this selective advantage. The native mutant cells die out and the transplanted cells take over and repopulate. That's how this really works. This is FAH activity in our pigs. Essentially, there's no FAH clearance, where in wild type pigs it's removed quickly. This is our first litter of these 3 pigs. At 1 month, they've been on a protective drug, NTBC. When you stop NTBC, this guy, they just stop growing and they develop liver failure. And over the past 2 years, we've studied 3 groups. One group on NTBC that essentially survive 100%. You don't give them any NTBC, they're dead of liver failure by day 21. You treat them with NTBC for a month and then stop it, they have sort of a longer death. But it's a very similar pattern to the human disorder. There's a few subtle differences, but basically this is a very good large animal model of a human disease. So it has applications I don't have time to talk about, but one of them is to take this mutant pig, do a liver resection, which we do in my lab. We digest the livers and do gene correction ex vivo, so it's not in vivo gene therapy where there have been deaths. This is correcting the cells in a petri dish outside the body and then transplant the cells back in. So we transplant about 2 to 5 grams of hepatocytes, and after 6 weeks, the liver looks like this, and these red cells are corrected pig cells that have been auto-transplanted that have now expanded. If you do a math analysis, our 5 grams of hepatocytes are now 150 grams of hepatocytes that have expanded over a few weeks in vitro, and the cost is essentially feeding the pigs to grow all these hepatocytes. So this is a proof of concept that you can regenerate a pig liver with hepatocytes. These are still pig cells. This is not human cells in this slide. So the third is, how are you going to transplant human cells without rejecting human cells in a pig? My first idea was to follow the fetal tolerization idea of Peter Medawar. He won a Nobel Prize for this. I suspected it worked. And it does. We've published a paper that you can get tolerization of pigs by injecting human hepatocytes in utero. So on day 40 of gestation, a pig's gestation is 115 days, so right at the end of first trimester, before T cells and B cells start to form, we do a in utero transplant into the 15 to 18 piglets, fetal piglets in this litter. We exteriorize the uterus, we use an ultrasound probe, inject. The liver is actually a pretty big target in these day 40 fetal pigs. Inject our human cells. We've injected a variety of cells, iPS stem cells, all the way up to hepatocyte-like cells, to mature hepatocytes, to figure out what the best cell to inject and for tolerization. We've used a variety of doses. Little piglet born, and then we study them for at least 8 weeks to look to see if the human cells have expanded. This is our first, Pinky was our first eye cell, so this is an iPS-derived hepatocyte that we injected in utero in Pinky. And at birth, this is an ELISA that shows human albumin in his blood. His litter mate that we didn't transplant had no human albumin, his mother had no human albumin. So he was born, we looked in one of the other litter mates that had been transplanted, his liver, we found nodules of human hepatocytes that were there at birth. Wanting to see these liver nodules expand. Unfortunately, they haven't expanded in our single knockout pig. This is the immune response. This is a normal immune response of pigs to human cells. Very high response. This is week 8. So they're not born with a response, but they develop it over maturation after birth. So this is a sort of after birth phenomenon. In our fetally tolerized pigs, essentially we've eliminated 95% of this response, but there's still a little residual response that I wasn't sure what to make of. One of my immunologist colleagues helped us develop this ELISpot assay, which will let you measure. The first assay was an antibody response assay. This is a better cellular response assay. And you get a very strong response. Fetal tolerization reduces it, but it's still higher than baseline. Essentially, we think that there's some rejection. Fortunately, 2 months ago, there was a report of the first SCID knockout pig by the group in Missouri, and they essentially knocked out the RAG gene, which is a T-cell differentiation gene, injected human iPS cells, and got mature teratomas to form in these pigs. This is what their figure from their paper looks like. So there's endoderm, which is where hepatocytes would come from, mesoderm, and ectoderm. So the current plan, which we're working on, and while I've been between the meeting, I've been writing an NIH grant to fund this, to create a double knockout pig that'll have both FAH and RAG2 deficiency. When we started this project, nobody was knocking out genes, and pigs was very difficult. Well, in the past 2 years, this field has just exploded, and now with CRISPRs and TALENs, you can do these things very rapidly. It's almost a no-brainer. So I take that back, and nothing's a no-brainer. But so that's where we're at right now, is to create a double knockout pig that's immunodeficient with FAH deficiency. So this is our first generation system that we're going to the FDA with to use pig hepatocytes in a bioartificial liver, and then the future generation will have our genetically engineered pig to mass produce human hepatocytes. You might ask, why do you need both? Pig hepatocytes are going to work fine, in my opinion, for about 2 weeks, and then there'll be an immune response that's developed. So that's where, if you want to do chronic long-term therapies, a humanized device would be better. This is a photograph I took 2 years ago. We went to Everest Base Camp, so I'm on top of Kalapatar looking across at Mount Everest. Base camp's down here, this Khumbu icefall. So my analogy to our project is like an expedition to the summit of Everest. You know, we're down here, we've got our device at base camp. We've actually made it through the Khumbu icefall, which is one of the most dangerous parts of the trek. Up ahead is safety trials, a pivotal trial that both these require lots of money that we're trying to raise, with the goal of an FDA-approved device at the summit. This is the Sherpas on the route, the people in my lab that are doing all the work, that are there late at night, you know, that week of the treatment. And thank you very much.